discussion or a single side discussion? बताइए. What do you want me to do? You want me to give an interactive lecture or one side lecture? हाँ? It should be. जवाब ही नहीं आ रहा, पीछे से नहीं आ रहा. मेरी रिकॉर्डिंग हो रही है, जल्दी बताओ. You want to do the interactive session. Interactive is that sometime in between I may ask for the response from you all, right? Right, because without your participation I cannot make it interactive. Is that okay? No? No? Okay. 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 So very good afternoon to all of you, and as a duty has been given to me that an in silico tutorial for computational approaches in drug discovery, right? Tutorial means definitely it's not a teacher role. Tutorial में हम during our classes in our graduation and in the post graduation term, when we used to run the tutorial, it is by the students, like we flip the classes, right? So you should be on this side and we keep on discussing, we keep on giving the inputs what the student is discussing, you know. Now under this umbrella I have given the topic that computational approaches are accelerating the drug discovery research. But before that I would ask you, anyone or who one is knowing the meaning of drug discovery research? Anyone? What do you mean by the drug discovery research, if I ask you? Anyone? Okay, at least one response I expect from the audience. Anyone who can tell me what is a drug? At least one response from the audience. What is a drug? So drug, now let me you agree with me or not, all the drugs are chemicals, but all the chemicals are drug. Which statement is true? All the drugs are chemical, but all the chemicals are not drug, right? If I ask you, definitely you must be, this audience sitting over here would be of the different ages ranging from 20 to 25, and in the journey of 2025 you must have come across with any one tablet, a very one common tablet, paracetamol, Disprin is there, painkiller, diclofenac is there, ibuprofen is there, you must be using. So definitely it is a chemical, but how does a drug differ from the chemical that makes it different? So a chemical is called a drug when it interacts with a particular receptor or the target. Is that okay? Right. If any disease is there in the market, if I say that I want to, disease is what? Any abnormality of the imbalance, any abnormality or the imbalances of the hormones or the biological conditions or the physiological condition is resulting into a condition of the disease. Now I want to cure that disease. For that disease I said I am commonly using the word that I need a drug, I need a drug. Now what is drug? I need to discover a drug. What is that? I need to discover a drug. Now in our pharmacy there are two steps, drug discovery and then drug development. Anyone know the difference between this, drug discovery and drug development part? So this is a picture. When any disease is in their market, if I being a pharmaceutical chemist, if I being a medicinal chemist, I want to design a drug, I want to provide a cure for a particular disease, I should know what is the disease. You people are biophysics, you people are from pharmacology, you know that what is the difference between the physiological condition and the pathological disease. Any disease is generally like why it is happening. Until unless you don't know the reasons, until unless you don't know why the symptoms are happening, until unless you don't know what is the biological pathway or the target is being involved, you won't be able to design a particular chemical that is possible to go to the site and interact and it give a therapeutic response, then it is called as a drug. Is that okay? So journey starts from identification of the disease. If any one of you in the we people are into drug discovery, I am a medicinal chemist, so my area is to provide a solution for a particular, I am not saying I am the direct person, it's a collective science, right? I will show you one picture where how the journey of the drug starts, from where the journey starts. So the journey starts from if there is any disease in the market. Now try to find what are the reasons, what are the symptoms, what are the possible pathways that involve, what is the target for which I want to design the drug. Then medicinal chemists try to find a suitable chemical that got the tendency to act on the receptor. Then it would be tested on the animals, followed by preclinical phases, phase 1, phase 2, phase 3, and I hope you all know that what are these phase 1 and phase 2, phase 3. Depending upon after the successful results in the animal, we put it and give it to the healthy volunteers to take the toxic effect. Then we check it on the patients with a smaller number. Then we give it to the bigger number in the hospitals. Ultimately, we go for we find the investigational new drug form, and ultimately, when this part is there, till the molecule is being discovered, chemical, it is called the drug discovery process. But conversion of that chemical into tablet, into the syrup, into the injection or a suitable formulation that it can be given to the patient, that part is called the development process. Is that okay? Fine. Now drug discovery, right? You might have heard about the quinine. You might have heard about the quinine. Quinine is natural or synthetic? Natural. So the thing is, the journey, we chemist people are like randomly, I will come to you what is this terminology is called when all of a sudden you people are starting any new research. But for all of us, very basic research that is being inspired from nature. Okay, so quinine, it was around 1837 when the 400-year back when the cinchona bark was used to treat the malaria. Are you aware of this fact? 400 years back. Then it took 200 years to discover that quinine is present, to isolate the quinine from the cinchona bark, to discover, to establish its structure and to establish its mechanism, and ultimately after 237 years, how many years? 237 years. By that time, the person who must have discovered this, he might have died, right? But he has given a blessing to all the humankind. Is that okay? So just note down the journey. So drugs are nothing but, if you look into the market, all the drugs are small molecules, and maximum 99% of the drugs are organic in nature. Okay? Then they are complementary in shape to the target. This is the very important. If I have designed certain molecule and if it is not fitting to the receptor, this chemical will remain a chemical. Important is that a chemical, to show a therapeutic response, its complementary shape to the target is very important. Is that okay? I always make it understand in the form of a lock and key. A lock will open until unless there is suitable key to give a noise. Is that fine? Okay. So oppositely charged. If I say that here is a molecule and here is a target, now it's okay, they are complementary in shape. Shape में fit हो रहे हैं. But what about the charge? If they are both are the opposite charge, will they attract each other? No. There would results into the opposite. So that is the basic fundamental of a drug, is that any chemical that got the tendency to interact with the receptor, that is complementary in shape and opposite in charge. Now in your terms, what are the targets? It could be enzymes, you people are working on the enzymes, it could be biomolecule, it could be ion channel, it could be transporter, DNA, RNA, and I think audience sitting over here are exploring the different fields. So you all are working under the different, someone is working on diabetes, someone is working on the neuropharmacology, and different concepts are there. Is that okay? Now, so nature is the one that inspires us, but problem is that if I cut, I hope somebody of you sitting in can imagine that if I cut one plant of kilos, I can get the amount of the chemical quinine in milligram level. Now in order to serve the nation of 140 crore की हमारी population के अंदर है, what do you think? This way I would be able to remove the gardens, I need to clear all the medicinal gardens, it would be free of the cinchona. So that is not the solution. So nature inspired me that quinone is present in the cinchona bark. Now in the chemistry lab we need to find out the solution how quinine can be synthesized. And do you know that what is the better version of the quinine that is being synthesized, that was synthesized in the lab? Anyone? Chloroquine, hydroxychloroquine. So these are the two examples that were synthesized by the chemist in the lab, but after the inspiration from the nature. So this is how the journey of the drug goes. Now drug discovery, no doubt nature is the one of the biggest source, right? But we cannot use the nature, as I told you the resources are very limited, okay, right? And other approaches is that penicillin by serendipity, right? By chance discovery is there. One microbe, say we extracted out the penicillin and that penicillin was used to kill the another microorganism, that is one of the approach. Another is that screening wide variety of the drug obtained. Artemisinin, again artemisinin, I hope you know that it was around 1967 when Dr. Y2 from Japan started working on the 2000 herbs, and then ultimately he reached out to the 640 herbs that show the potential in malaria, and then it was around 2006 when he got the artemisinin isolated, and 2015 he got the Nobel Prize. कितने साल की जर्नी हुई? 50 or 60 years. So 50 to 60 years, like starting from the nature, then find the structure establishment, then mosquito role, mechanism establishment, and hold this to get into the FDA approval, formulation development, and after all ultimately acknowledgement in the term of the Nobel Prize. It took 60 years of the human life. Fine. Chemical modification, मैंने अभी आपको बताया quinine, chloroquine and hydroxychloroquine, they are the modification. Quinine was a nature inspired. Chloroquine and hydroxychloroquine, they are the synthetic modified derivative of the quinine. Then combination of the two drugs. Sometime, now the malaria is one of the example. You might have learned about ACT, artemisinin combination therapy, right? Malaria, when the quinine was being used, mosquitoes getting resistant, or they are getting immune, we are getting like mosquitoes were getting resistant to that, right? So that was an alarming situation to develop new drugs. Artemisinin came into it. Artemisinin is having a short life, but quinine was having a long duration. But in terms of potency, artemisinin was more active, quinine was less potent. So this gave a new thought, okay, let's combine the two drugs. And this is also drug approach, yes, in order to cure any disease. So they thought to let's combine the two drugs that will act in a two different way. So that's why number of the diseases are presently being cured by giving the two medicines rather than one. And last one, where we are turning around, rationally drug designing. Now when we were at your stages, like long 20 years back, when we did our PhD, we used to go to the literature in the chemistry lab department. I remember we used to go to the chemistry lab, and in a one day, single day, we hardly make it possible to check one journal. Like if I am working on the prostate, hardly in a one day, if any, like let us say 2024, 12 volumes are there. I could make it possible to check 12 volumes only. To check prostate is there, prostate is there, prostate is there. Manually we used to do it. But now you people are just a click button away. At that time, we keep on synthesizing the molecules without knowledge, because we were not knowing the receptor structure. So we kept on synthesizing the molecules, let it say 100, thousands, and then in my 5 years, because practically let admit all of us, a PhD is journey of 5 years, right? If any one of you are working on any one target, at least in a 5 years, you can explore one pathway. Is that okay or not? Right? It's not possible. One particular disease can have a number of pathways also. But in a given period of span, you can manage to study one pathway or one parameter or 2 to 3 parameters. So we used to, at that time, we were not having knowledge about the receptor, we were not having knowledge about the targets. So we kept on synthesizing number of molecules, and my junior will come and he will then synthesize, or I would be submitting my sample to the CDRI Lucknow or to the any another institute, pharmacology people, okay, you please test on these molecules, and after 5 or 10 years he will give me the some outcomes. So a timeline. Now reverse is happening. After 10 years of spending, I come to know that my molecules are not working on that receptor. So what is happening? 10 years of my life, 5 years of my life, no doubt I have been awarded my degree. But if I talk about the success, how success is being defined? Success is that how the society is being benefited. Okay, no doubt every observation in the research makes a big contribution. There is a journal of negative observation also. Please always never drop your idea, because if I have worked, I have spent my 5 years of research on those molecules, it means I am passing my information to the coming generation, don't work on these molecules. If I would not be doing the publication of the negative results, people will start again doing the research on that part. Okay, so the rational drug design is that I would be starting rationally, logically, after knowing the structure of the target, what is the protein structure, and how this is how now we are stepping into it. Now look into this picture. I would ask you at the last, how many molecules we have synthesized? 10,000 or 1 lakh molecule. After 1 lakh molecule, hardly 1 or 2 molecules make it path to reach to the market. 1 lakh molecules are in the lab. I am talking about the in the lab, and it's not possible for me to synthesize 1 molecule, 1 lakh molecule. For researcher like me, who all are working in the area of the prostate, they must be working in the different area in the prostate, but different skeletons. Now I have combined the information from all these molecules. Let it say this data has been collected, and do you know what this data is called as? If I am collecting the chemical information from all of you, let it say you all are medicinal chemists sitting over here, and you all are working on 10 different 20 skeletons. You all are synthesizing different molecules. Now I am keeping this pool at one place. What is this base? What is called? Yes. What is this called as? Now it's a database. Database. Kiska? Chemical ka. So chemical database. Same way you people have the database also. Kiska? Biological database. Don't you have? There is a target proteins. बहुत सारा है. Database क्या है? Right? It's a collection of that. Na? Google is one of the biggest database these days. Perplexity is there. Chatbot. Number of the resources are being there. Now look into the success rate. After like 3, 6, 5, 10, 1, 2. Overall it's a 18 years of minimum 18 years of journey is required for a chemical to become a drug after spending $1 billion of the dollars on it, right? And you cannot deny that you all are using the animals for bioassays. Maximum. We medicinal chemists may or may not use the animals, right? We can skip that part because our part is to synthesize molecules. But those molecules hold value until unless they are not being tested on some animals, they are not having any therapeutic value. In order to check therapeutic value, definitely I am using the animals. You people are using in order to study the in vitro, in vivo, to check the therapeutic efficacy, in order to study the whether they are physiological, in order to study the what is the fate of the drug in my body, then bioassay, preclinical toxicity, for any kind of like studies you people are using this. And yes, same way you cannot deny that somewhere we are torturing the animals. We are torturing. I am not saying, we also did it and we have to do it to some extent, but is there any way that we can curtail it on it? Yes. So in the laboratory they are deprived, sometimes we make them deprived of the food, sometimes we are giving some injections to induce the disease. This way it means the animal is under pain, under distress, and in order to perform, like I have said that we need a skilled person. I am a medicinal chemist, I am not the right person to do the animal study. I would be in the need of the help from you people. So there are certain issues while handling the animals. I am not saying that we can. Neither computers can replace the humans, nor the computers can replace the animal studies. But there is a way. We need to find the alternative. I hope you have heard about this 3R strategy. R means reduction, refinement and replacement. How we can reduce the number of animals? How we can modify the biological method that it gives the less pain to the animals? Is there any alternative that I can use and I get the same observation of the results? And the answer is lying in the. So I hope you are in vitro models, you know that. In vivo models, you know that. Now the biological evaluation using the computers is called the in silico. Here the two branches joined hand together. What type of two branches? Computer sciences and the chemistry people, or the computer department and the biological sciences. Without these two, computer science, biological, stats, math, algorithms, everything is being required. I am just giving my data to the computer peoples, nothing that, beyond that. Computer people does not know much about the benzene structure even now. For them benzene is what? 01010101. But how that benzene is being drawn, that is different for them. But I can see only 6-member ring with 3 double bonds. It all depends on the perspective, how he is looking and how I am looking at that person. Okay. So it is the integration of the computer people and integration of the biology or the chemical or the stats or the physics. All the integration of all the branches is being required. Right. Now I am giving my data, I am collecting let me say all of you people are collecting your data, you are working. Now you people are providing this data to the IT people. IT people are storing that data as a databases. Now again the next generation will come, it will put some keyword into it, it will try to retrieve the information and try to utilize this knowledge into your field. So this is how implementation of the integrated approach, that is called the in silico tools, data is being stored, information is being extracted out and you will apply your knowledge. Don't leave everything on the machines, don't leave everything. Okay. So you need human integration is always being involved, always. If you will say ki let do the machine do it, PhD degree he will get it. Okay. So human intervention is always being required. Computer sciences, chemical sciences, biological sciences, physics, stats, math, all are working together. All are working together. Right. So this is how integration of this approach will help you to minimize the mistakes, to make the faster decisions, and in order it will reduce the time to reach to the market. I will show you how it becomes. Initially I show you the slide where the 1 lakh molecules were being synthesized and hardly after 18 years 2 molecules were reaching to the market. But after the computational from past many years, since it has driven into the market, around 45, after 45 molecules, 23 molecules are showing success, and out of 23, 5 molecules have already gone into the market. So can you compare the 1 lakh molecule versus 45, 2 molecule versus 23 and 5 molecules? Success rate, if you look into the percentage, it is the 5 molecules over 45, that was 1 over 1 lakh, right? This is how the computers look into. But आप लोग क्या देखते हैं? बटरफ्लाई. आपको क्या दिखाई दे रहा है? आपको 01 दिखाई दे रहा है या फर्स्ट आई में क्या दिखा रहा है? वेरी फर्स्ट आई. इल्यूजन इमेजेस आती है ना, इंस्टाग्राम पे आती हैं, what your eye sees was. So we being from the science people will try to have a look on the mosquito rather on the 01. I'm least interested in how the 0101 is being displayed. But what is catchy to me, to my eyes? It is the bacteria. So this is how benzene structure, chemical structures, like it is growing field, algorithms, mathematical instructions are required. Researchers have integrated techniques, artificial neuronal network, formal logics, mathematical stats, operation research, economics. You cannot say that. You cannot skip the psychology, physiology, linguistic, neuroscience. All fields are working together. All fields. If you really want to get the best use of it, so it is the one of the biggest two. So computers increasing, computers are not restricted into the, like you people are using on your mobiles, you are using on the Instagram, Facebook, like weather forecast estimation is there, number of that booking hai. You people are using the technology, or you would like to say you people are living the technology? It's a better statement that you people are living the technology. So technology, like it has gone in, it has delved into the number of the applications of the computer in the healthcare. You know that disease identification, CT scan is there, it's computer. Doctors are keeping the records, it's computer, right? Smart health records, medical imaging, then drug discovery, manufacturing, disease predictions, everything. I hope you are aware of the, like China recently 10 days back has introduced the AI machine in the market. You just stand in front of that machine, it will diagnose your body and it will give you the blood reports and all the tests that what kind of disease you are having. Are you aware of this technology? 10 days back. And I hope also you know that China has introduced the hospital with 40 robots where no single doctor is there. So what are robots? What is that machine? Ultimately it is IT, and IT is what? Computers. You can use the word AI or you can use the word computers, but working it. But the thing is, how that machine is working, how it is able to, if you look at, you stand in front of the machine and it will help you to diagnose the, like somewhere data has been driven, data has been added. Based upon those algorithms, it will try to predict your disease. Algorithms तो लगेंगे ही लगेंगे, data अंदर जाएगा ही जाएगा. Data is important, integration of all the branches is important. If I say that open your mobile now, right, and just Google out single word paracetamol, how many pages you would get? Hurry up, anyone? Kitne? Just I can tell you, right? It will give you approximately 1,20,000 pages. Honestly admit that how many of you have explored second page? No. We go look at the first or the first pages, and 20 pages, अगर 20 sheets आती हैं पहली sheets पे तो हम 20 को ही देखते हैं. It's very true. हाँ, filter लगा लीजिए, आप फिर भी चले जाइए. Still you don't have that stamina to go and look into the 1,20,000 pages, right? Now that paracetamol word is there, right? Your interest is of the paracetamol. Now from where this 1,20,000 pages are coming? I am chemistry person, I would be looking into the synthetic approaches. You people are pharmacology people, you will try to look into the, like how it is acting, right? Doctor is looking ki what kind of the latest information, side effects are there. So everyone is working on it. Biochemistry people will try to look at another way. The same 20 pages are there, but everyone will try to look from its own angle, right? So अब उसी को देखने का, when after finishing this class, I will show you one more dimension how to look into that page. Okay, so this is like how the computers are entering and keeping a hold on our daily lives. So computational tools in the drug discovery process, it has become, need evolution is not a process force, ज़रूरी नहीं है, but it has become the part of the journey, right? We are not saying to use it, but over a period of time you need to apply it, because FDA guideline has now been approved to use the computer predictive data. I will show you that slides also. Slowly I will come back. So high, because this use of this computational tool will high attrition rate. Attrition क्या होता है? Failure or success rate? Increased expenditure. It is being required to control, to address the issues of high failure rate. Attrition means failure rate. I was synthesizing 1 lakh molecule. Now after use of the computer, if I am synthesizing only 45 molecules, don't you think my expenditure, there will be a curtail on my expenditure? Yes, there will be curtail. Increased volume of data, definitely volume मेरा data बढ़ जाएगा. Increased globally requirement. ठीक है. Requirements, abhi recently, if you might have seen that one of the syrup has been banned by the FDA. Cough syrup is there. ठीक है. So the moment any side effect is there, FDA come into picture and it immediately banned. उन्होंने पूरी पंजाब में बैन कर दिया है उस सिरप का यूज़ करना. Why? यूज़ करें. तो रेगुलेटरी रिक्वायरमेंट्स जो होती हैं वो टाइम टू टाइम दे कीप ऑन चेंजिंग, राइट. सो चैलेंज इन द टाइम, द बिगेस्ट इज द टाइम प्रोसेस. If COVID is over here, if I would say that I need to discover a drug, and in order to discover a drug I am in need of the 3 to 4 years or the 10 years, by that time world would have been swept out. Agree or not? Anyone interest, anyone can tell me COVID की वैक्सीन को इंट्रोड्यूस करने के लिए कितना टाइम दिया था? Yes? No idea. क्यों? ड्रग रेगुलेटरी तो कहती है कि मिनिमम आप 15 to 16 years clinical trial. So that was the regulatory decision on immediate pandemic basis. Otherwise they are not allowed, supposed to release the drug in the market, right? And how that drug was, how they have discovered the drug that can control the COVID? Again, that was the computers. Okay, because you people are from different field, I will try to make it interesting by showing you the one example that's true from the COVID, जिससे हम सब affect हुए थे accordingly. Okay. So now here is the drug development. Earlier I showed you this slide where the traditional conventional ways were there. Now look into disease से लेके clinical trial तक, all the phases have been taken up by the computers. What are these terminologies? Bioinformatics. Informatics, what do you mean by informatics? Bioinformatics, it is the integration of the IT and the biological data. Cheminformatics, integration of the chemical and the IT. Reverse docking, protein structure, target, library docking, de novo, pharmaco, QSAR, structure based, in-silico ADME, physiological. So these are the all computational tools and the approaches that are being in trend to fasten up. If you look, remember my first slide, computational tools are accelerating drug research. Accelerate क्या होता है? Fasten up. They are helping me to fasten up. So all these tools are. It's not like you can use maximum about 2 to 3 approaches you can apply your during PhD projects, right? And sometime if you look into the, from today onwards, maybe the vision you get changed, in silico, in vitro, in vivo correlation, like what you have predicted from computation, try to correlate with the biological data and then try to establish a relation. So this is how it is called the in silico in vitro correlation. And sometime medicinal chemists use it in a vice versa, designing synthesis and evaluation. In our time we used to say it was synthesis and evaluation. We used to keep on synthesizing molecule and we keep on evaluating her. But now the concept is designing, synthesis and evaluation. Now what is the designing? I am designing the molecules hypothetically on the computer. Then I would be testing it on the computers. Those molecules that will be showing the good results on the computer, I will pick it into the lab, from dry lab to wet lab. Journey is called dry lab to wet lab. In silico, in vitro correlations there. So you might be, you people have seen number of paper, but शायद हम सुनने के बाद वो और comfortable हो जाता है. Okay. So these are the various tools, and it is will be very tough to cover all these tools. But let me try to give on the structure based drug designing. This is the one of the tool that you all may need over here. Structure based drug designing. Now structure based drug designing is the tool किसकी स्ट्रक्चर? Whose structure? If I ask you, very good. It is the structure of the receptor, right? If now what according to you, what you all are biological people with a background, what do you mean by the receptor? Target. What is that?
Protein.
It's a protein. And if I have further break down the protein, what is protein? It's amino. If I further break down the amino acid, what is that? No, my dear. Look into the chemistry part. It is a carbon. It's having one H, then amino group, carboxylic group is there, and one R is there. So one amino acid differ from the another depending upon what R is there and how they are the उसकी stereochemistry कैसे होती है. Is that okay? So that is the amino acid. Now your genomic structure, how it differ from each other? आप लोग बहुत ज़्यादा बायोलॉजी के पर्सन हो, हाँ बताइए. How my features do you differs from you? How? Genes, यही है ना. Genes तो वही है, basic तो 4 bases तो वही हैं. Then still how we differ 20 के 30 people and we don't matches with each other. It's a rearrangement, right? Now if you work on any target, you pick any target that comes to your mind, जो भी आप जिस फील्ड पे आप काम कर रहे हैं, उसकी कितने अमाइनो एसिड्स हैं? Anyone can guess? Rough idea? किसी का रिसेप्टर हो? नहीं पता? कितने? 51. Now do you remember the sequence? फिर? So how it will, don't you think it is important? Now you people know that protein is like you people draw the structure in a one dimensional way. What is one dimensional way? Amino acid, amino. Now two dimensional way, then 3D, and in your body it is present in a 2D, 3D, 4D, what? 4D. Right? Don't you think the folding of the protein and the sequence of that amino acid makes a difference? So it means if I know the structure, if I know the sequence of the amino acid, how they being are folded within my body, will it help you to design your molecules? Will it help you to design your molecules? Now what is this called? This is called kiski structure. So that is called the structure based drug designing approach. You know the defining the binding geometry. If you remember my first slide, I told you that drugs are nothing but the small organic molecules that are complementary in shape to the receptor and that would have the opposite charge. So it means if my amino acid is having NH group like that and the carboxylic group is on the back end side, so it means while designing a molecule, don't you think if I am having prior in hand information, what kind of the groups are present over here, can I introduce that kind of the groups that immediately? So what I am doing? I am defining the binding geometry. So this is called the structure based design. And here you people, this is one of the biggest people for the field for the biophysics people and the biochemistry people. You people are required here. You know that the chemistry, I am not technically sound in this case of this light. You know that the protein structure establish करने में आप लोग, I think you people are playing a big role in that. You know the chemistry, उसकी resolution चाहिए आपकी, right? Sequence is important, resolution is important, NMR data is important, right? IR, X-ray crystallography is important. So you people are doing that. You people are developing the machines. You people are establishing this high resolution requirement is that. Now 50, he said कि like ma'am insulin is having 50 amino acid, but he doesn't remember the amino acid sequence. But computer does. Not only computer does remember 50 amino acids, but he can remember like 256, let us say if any receptor I am working, it is working on the 250. You people have come across with the different isoforms of any enzyme. You know that? There are isoforms of the enzyme, right? What is the difference in the isoform? How you define the isoforms? COX1 and COX2. You might have heard about it. Anyone? What do you mean by the isoforms of the enzymes? How they differ in the different? Yes? सेम जीन नंबर उनका एमाइनो एसिड सीक्वेंस सेम रहेगा, उनकी वर्किंग कंडीशन, उनकी प्रेजेंस. लाइक लेट इट से इफ आई एम वर्किंग ऑन वन एंजाइम, इट इज प्रेजेंट इन माय बॉडी in male human body, it is present at the 3 different stages. One is present in the prostate, one is present in the skin, and one is present in the hairs. So don't you think if I would be designing a molecule, if I design a molecule for the prostate, there is the information regarding the enzyme present in the prostate or on the skin or in the hair matters? Will it increase the specificity or sensitivity? It will increase the specificity. I am designing the molecule specifically for the prostate. So it means these are isoforms. Is that okay? So this is called. So pocket topology. Now how this isoform, the isoform present in the prostate is present in a different conformation, that present in the hair and that present in the, so that is called the pocket topology. Okay. The molecule that can interact with the receptor present in the prostate may or may not be, should not be able to interact with the, it should or should not? Should not be able to interact with the receptor. For that what is important? Pocket topology. For that what is important? High resolution, conformational state. So well defined pockets are there or the disordered pockets are there. So computers are continuously helping you to predict, to give you the data on daily basis. Earlier times mein you people are, this is, I am really telling you that number of jobs are waiting for you in this area. For go to the LinkedIn and looked in for the jobs for the SBDD or the computational tools or the chem informatics people. You people can help and enter and take the LinkedIn pe jaiye and number of jobs are waiting for you. But only thing is you need to just do the some small AI, कोई भी 1-2 महीने का कोर्स कर लीजिए and you can do it. Okay. So well defined pockets and yet the information is being given. Another is ligand based drug designing. Okay, I will come it on later end because it may not that help you again. So you might have heard the word docking. So structure based drug designing is nothing but the docking. Structure based drug designing is nothing but the docking, right? Simple ways, I will always try to make it very simple way, lock and key. If you are having 20 keys and 1 lock, by looking into the keys, can you make me understand कि किस type की key लगेगी lock को? I hope you can. क्यों? How come? If you are having a bunch of 20 keys and there is a 1 lock, can you filter out 2 or 3 keys based upon just looking at the lock and the key concept? Yeah. By size, right? By a roughly idea about the pocket, by looking. So now I will take back to you, the lock is here your enzyme, that hole on the lock is called the pocket and the keys are the chemicals. Is that fine? So now this docking is a paradigm shift in the drug discovery. It is being, it's not simply merely like, it is being used by many ways. Polypharmacology, drug repositioning, virtual screening, targeting phishing. Let it say if you have one molecule, but number of the, let it say 230 receptors are present there, right? Databases में से, this is called bioinformatics. Databases, PDB databases or the biological databases or the protein databases. If you have 230 proteins are there in the database, but you have one molecule, आपका बहुत प्यार है उस molecule के लिए. You said I want to do something for this molecule, right? Now what you will do? You will try to, this is called the reverse docking. You will try to check the interaction of this molecule towards all the 230 proteins. And with the best interaction, you can say this particular molecule got the potential to work particular against particular type of disease. Are you getting my point? Is that okay? So that is the target phishing or the profiling or virtual reverse docking भी हम उसको बोल देते हैं. Prediction of the adverse drug reaction. Now if he says that insulin, I said कि ma'am I want to do the like let it say any I said कि insulin के लिए काम करना चाहते हैं. He knows that my target is for insulin का जो भी इनका receptor है, right? So molecule should interact with that particular receptor, right? But I am checking, I am putting the model while evaluating or while doing the PhD. I have tested my molecule only for that target. How you can be sure it is not working for another targets? बताओ. मैंने अभी बोला कि PhD में प्रैक्टिकलिटी रहती है 5 years की प्रैक्टिकलिटी कि PhD करनी है 5 साल है, लेकिन उस 5 साल के अंदर आपके पास एक पाथवे होता है, राइट. दैट वाज द बिगेस्ट क्वेश्चन टू मी ड्यूरिंग माय PhD वाइवा. I always share this. It's an inspiration for everyone. He said, I said कि my molecules are very potent for the prostate cancer. He said कि okay, how come? I said, sir, I have tested them for the prostate. So he said, did you test it for other cancers also? मैंने कहा no. So then it means in order to ensure that whether it got the potential to work on the other sites also, it means I need to check on all those also. Now this can be, it means I need to check, this is called the polypharmacology. Polypharmacology, not exactly polypharmacology, I may present it. Now this particular molecule, if it is showing, let it say I have checked it for the lung cancer, colon cancer, skin cancer and the prostate cancer. If it has shown very good activity against only the practically in the prostate cancer, it means it's really good. If it is showing some of the activity against other cancers also, then it means it got the tendency to work on the other targets also, right. Now effect on other targets may be desirable or non-desirable. If it is non-desirable, then it means you are checking the drug interact with toxicity effects. Undesirable effect is called toxic effect. Let it say if I want to do this study in practical on humans, on animals, it may take how many years? Whatever the period I have shown. But all this you can do it by using simple computers in 1 month. So adverse drug reactions, polypharmacology, he said I want, let it say you said ki, I said ki 5 alpha reductase or the enzyme is present in 3 forms. I am in my 5-year tenure, it is possible for me to check only one pathway. But my examiner, my reviewers are asking, have you checked it for the another 2? It is not possible practically for me to do all this study. Definitely next junior student will come and do that. But by giving the predictive data, doing the studies on the computer, I can give and defend myself that yes, I have done the computational studies, predictive studies are there, my molecules hold the potential, and that's why we can do for it. Is that? So polypharmacology means if your molecule got the tendency to act on the number of the targets rather than one. Now the action on these additional target may be desirable or undesirable. If it is undesirable, then adverse drug reaction. You may do the target fishing. Fishing में क्या होती है? Receptor. Fishing क्या होती है? You pull the right and pick out one fish. So it means if you are having one hook, that hook is your molecule, and amongst the 230 receptor, your receptor has shown the activity against the one. It means you start your journey with this molecule against that particular receptor for a particular disease. So this is how virtual screening. I will show you one examples. Drug repositing. Repositing मतलब क्या होता है? Any idea? Yes, Shabash. Yes. And any example for this? Very good. वेरी गुड. किस चीज़ के लिए? वेरी नाइस, वेरी नाइस. Yes. So like drug is already. Do you think this will help in saving the time and the resources and the toxicity data? Aspirin was there earlier, it was for the headache and whatever it is. Now it is being used as a for the blood के लिए use करते हैं. So whatever the data was studied on the patient, on the animals, on the clinical trial, the same data will because it has been studied for those. So repurposing is one of the again another attraction of the science and the research these days. Drug repurposing. अगर FDA साइट पे जाते हैं, FDA की साइट पे जाइए and you will look into that number of the 40 to 50% of the drugs अगर clinical trials में पहुँच गई हैं that are based upon the repurposing. Again by using the tool computational. Which tool? SVDD. SVDD means docking. Is that? So if you want me to explain, these are the various steps before doing that we need to prepare the protein, we need to prepare the ligand. It's very important to find out the binding site. Then we need to remove our water, or what is the importance of the water or not, right? Then molecular docking, and ultimately score is being done in the form of the like G score and so on. So these are the basic steps. Better it would be if I will be showing you one example from my lab. And also if I ask you, here different two type of populations are there, or you can say three. One is male and female, another is age factor के हिसाब से. I am in the age of 50, you people are at the age of 25, right? And there are geriatric, pediatric and adult and the young ages are there. If I want to if I want to sell a drug, if I want to bring a drug into the market, don't you think I need to study the drug for all these age factors? I need to study how the drug will behave in my body, right? Biochemistry people, I have synthesized, let it say 50 molecules, and on one molecule one student can do the PhD, because he will be doing the study how it is going into the body, different compartments, blood, urine, serum, like what is happening, what are the different metabolites, what is the journey, half life क्या होती है, कहाँ कहाँ travel कर रहा है. So there are the number of the way. So even one molecule के ऊपर I have synthesized 50 molecules in 5 years. Now from one molecule one PhD students can be trained, because it's not only the PhD trained, but the thing is, in order to bring that 50 molecules into the market, I have to collect the data based upon their absorption, distribution, metabolism, protein binding, you know that, right? So if the drug is getting metabolite, you know that. If I am having, you might have heard about that, someone doctor says that take this medicine with food, take this medicine without food. What is the reason? So don't you think again we need to look into this factor, that I am giving the, again I need to do the studies in the practically life way, right? So if I want to give this, like give the drug to empty stomach or stomach, right? It means food is affecting somewhere absorption, okay? This is one of the parameter. Now if a healthy volunteer is there and one is diseased person is there, right? Dose is different for a childhood, dose is different for geriatric, and dose is different for the adulthood. What is happening in the geriatric people? They are getting old, metabolism getting slowed down. Small baby is there, metabolism system has not developed yet. We are at the perfect age. So don't you think before, again once I will quote that, before bringing the molecule into the target, I have to provide all this data? Yes. Thousands and thousands of the papers are required in order to bring one molecule, and number of years and billion of dollars are there. Now with the help of computers, if you look into chemical spacing information, so a line ke left hand side pe is the input data, and the left and right hand side machine modeling is there that will help you to predict. This is called the ML and AL. You might have learned about all these things, you might have come across these terms. These are AL, hai na? Algorithms, and a machine, AI and a ML. So chemical specific information, whatever the scientists so far have developed different molecules, they are predicting the molecules or the data in the lab, and they are doing the physicochemical properties, partitioning behavior, tissue permeability. I am giving my all this data to the IT people, right? Plus I am adding the information about male, female, age, organ, kidney, liver, blood distribution. So this is physiological and anatomical feature. I am giving the power to the computers that this is the data, and this is the chemical space information, and this is the physiological. Now computer will do the algorithm and the calculation, and it will predict a model. Now you bring any molecule from your end and you ask the computer that based upon this model, please help me in getting the information, what would be the possible future of this drug in my body. And in order to do this study, I need minimum of 5 to 7 years. But a click away, and I will try to get it done now. हम इसके बाद हमारी एक छोटी सी एक्सरसाइज प्लान की है, जहाँ पे हम ये वाला काम करना है. We will be trying to predict the properties of the molecule. That would be my part. अभी हम जो tutorial में करेंगे, you will be doing that. So in silico I have provided. अब आप कहते हो मैम सॉफ्टवेयर है. सॉफ्टवेयर में क्या है? सॉफ्टवेयर में गार्बेज इन गार्बेज आउट, जो इन्फॉर्मेशन आप डालोगे बेस्ड अपॉन दैट मॉडल ही वो डेवलप करेगा. यू माइट हैव वर्कड ऑन सम मॉडल्स, किसी ने तो डेवलप किए होंगे ना मॉडल. मॉडल कहाँ से डेवलप होंगे? until unless I am giving the information or the data to the computer, from that information computer will try to develop the model. Based upon that prediction I will make out the predictions. I will make out. So chemical information, secondary drug screening. Very first screening is that, मैंने आपको पहला बताया था, very first screening is that this is docking is the very first. These days if you look into the any papers or into the docking is being used as a primary drug screening approach. Second screening process these days being implemented is that ADME properties. Okay, this is the oral. So I want to show that, let it say if you people are doing the in vitro or in vivo enzymatic assays in your lab. I am working with Dr. Tanzi, just like I have synthesized molecule. For me my research is done, because for 5 years I am struggling to synthesize one molecule. But from that one molecule, ma'am did so many of studies, like I am not in position to recall those pathways, sorry, I won't be able to do that, right? I am not the right person to comment on. So if you look into permeability, then transportation, melanin binding, ultra equilibrium dialysis, you people are doing PCR something, etc. So all there are tools, computational tools for everything are there, everything. Only thing is that you just Google out and ask for the AI or the computational tool for your study. I guarantee 101% you will get that tool for that. Some are free, some are paid. The difference between the free and the paid is that free में data updated नहीं होता. Let it say if she wants to do the ADME study by using the free software, क्या फ़ायदा फ्री सॉफ्टवेयर का? बिकॉज दैट इज़ नॉट द अपडेटेड वन. ओके, उसको इतना बेनिफिट ना. बट द कमर्शियल जो होते हैं, पेड होते हैं, उनके अंदर लास्ट मिनट तक का डाटा. सो इफ शी विल बी प्रिडिक्टिंग द इंफॉर्मेशन, if she is doing the research for PhD or really for the industry cannot afford the free softwares. Industry is paying लाखों करोड़ों रुपए पे कर रहे हैं for their softwares. Okay, so one molecule against ADME की वजह से in phase 37% molecules get fails in the ADME. So in our body on an average 40 organs are there. If I want to say that okay, you do the insulin, during insulin you are studying one organ, कौन सा? वो भी नहीं कर रहे ना. So it means in order to bring your research into practically into the market, so I need to, there are 40 organs present in our body and 400 cell types are present in my body, 400 types of cell, and FDA needs data on each and every organ, each and every type of cell before it comes into, before I can file the IND. 400 cells को study करना, right? Not at all feasible for one person, not at all possible for one institute also. Collectively we are working in different directions. But so there were these days computational tools are there and they are helping you to do this. Another tool is that homology modeling. In case you are not one, we have also used these tools, homology modeling. Have you heard this word? Have you heard this word, homology modeling? I said that SBDD. What is SBDD? Structure based drug. किसकी structure? If the structure of your receptor is not known and you want to develop what would be the possible structure of your target, then the tool is called the homology modeling. Homology means same. Model means I am developing a model. So homologing model will help you to develop the 3D protein structure of your receptor in case you PDB. What is the full form of PDB? Protein database. If the protein, do you have any idea how many यहाँ पे मैंने शायद लिखा होगा यहाँ पे yes, over 200 million sequence known versus only 22 lakh solved structures. मिलियन कितनी स्ट्रक्चर्स हैं हमें जो सॉल्व करनी है? हाउ मेनी स्ट्रक्चर्स? 200 million sequences are yet to be resolved, and how many have resolved so far? Only 2 lakh. Okay, yet so much of work is pending and you all can contribute to this, right? So homology content, X-ray, crystallo, Na, cryo, these are costly techniques, no doubt. In order to establish the structure of the protein, you need to isolate, you need to crystallize it, you need to establish its NMR data, and all this can be done by using the computational tool, and that is the 3D structure, that is the homology model. Dr. Tanzi, please let me know the timing. Shall I? 5 minutes. So I will be showing one case study from the lab, so they get interested. Okay. So let me take you to first. Yes. कोविड इंडिया में हमारे पीयू में कब लगा था लॉकडाउन? 17 मार्च. आई रिमेंबर डॉक्टर तंजीर और हमारा शुडेंजर लैंड किया था. वी पेड 6 to 7 lakhs दैट टाइम. द मोमेंट इट लैंडेड इधर से लॉकडाउन हो गया. डॉक्टर रिमल इज विटनेसिंग. 7 lakh we paid for this software, right? So I was getting continuously call from Schrodinger's head ki ma'am please do some studies for the India. China has already published 500 papers. 17 मार्च तक, बाय द टाइम जब तक हमें ये समझ आता है कि कोविड हो गया है, लॉकडाउन करना है, वी नीड टू क्लैप इट डाउन, वी नीड टू कैंडल डाउन द लैंड. दैट टाइम, बाय दैट टाइम चाइना ने 500 पेपर्स पब्लिश कर दिए थे बाय यूजिंग द computational tools, and it was the computer power only that they established the structure of the coronavirus too. It was the only structure. I think यहाँ पे मैंने लिखा होगा जनवरी 2020. जैन में 2020 में कंप्यूटर्स के साथ. Now what is this? This is the, if I want to bring the structure, if I want to design, COVID की वजह से number of deaths were happening, I want to have a control on the number of the deaths. What I need? I need a drug. मुझे stop gap arrangement चाहिए था. अब before that कि वायरस तो होल्ड में आ ही नहीं रहा था, राइट? वायरस इट वॉज नॉट पॉसिबल टू होल्ड. जैसे वायरस आ रहा था वो पर्सन किल हो रहे थे. सो द मोमेंट दे सेड कि ओके लेट कोरोना वायरस SARS-CoV-1 भी है आपको, इफ यू रिमेंबर, based upon the knowledge of the SARS-CoV-1, they predicted the possible structure of the 2, and they used the structure of this SARS-CoV-2, and they, please look into the figure. Look into the figure. 1.3 billion compounds. 1 पॉइंट किस टाइम तक ये पेपर आ गया था? 11 मार्च को. हम 17 मार्च को अंदर गए थे. 11 मार्च तक 1.3 बिलियन रैपिड आइडेंटिफिकेशन ऑफ द रेमडेसिविर का नाम सुना है? कहाँ से आया था? यहाँ से आया था. After screening 1.3 billion compounds after the possible predicted structure of the SARS-CoV-2, right, remdesivir was one of the, यहाँ पे I think ये रहा, hydroxychloroquine, PGI में hydroxychloroquine and remdesivir. I remember my one of the colleague was the very first COVID patient in Chandigarh. Dr. Gurpal was there, and he was being given the hydroxychloroquine, and that time it was given. So that is the success of the, that is the power of the computer. अगर हम वेट करते कि कौन सी ड्रग दे. नो डाउट रेमडेसिविर स्लोली इफ़ यू रिमेंबर इट स्टार्टेड ब्लैकमेल भी होना शुरू हो गया था. 25,000 की पुणे की एक सन की स्टोरी भी है जिसमें सन ने कहा कि प्लीज़ मुझे दे दो. एंड आई फील सम टाइम बैड, इट इज़ आवर जनरेशन ओनली, और या हमारी ही सोसाइटी के अंदर डॉक्टर हैं. ही सेड आई विल गिव यू एट द कॉस्ट ऑफ 2 लाख. Like rather than saving, it was a point of business for them. So remdesivir and hydroxychloroquine timeline shows 3 COVID pathway, and this was for the infographic. This was the outcome of the. And if I ask you the question, how the remdesivir was discovered, not discovered, like identified. यहाँ पे discovery की identification में फ़र्क है. Please note it down. I have not discovered. We have not discovered. What we have done? We have identified. From how many compounds? 1.3 billion compounds. वो compounds कहाँ से आए? From the chemical databases. And anyone knows the biggest chemical database? Yes? Biggest chemical database of free zinc database. If anyone wants to explore, number of molecules are lying there. Just category wise, skeleton wise, receptor wise, pathway wise, try to explore the zinc database. Zinc database. ठीक है? I would be giving the. So remdesivir was identified based upon the knowledge of the target. So that's why it is called the target based drug designing. I hope whatever the information I have tried to share. Yes, these are some of the pictures. Computer models are replacing the animal testing. We know the facts. FDA has already given the guidelines. Please give the predictive toxicology data rather than putting the animals or the humans on the stress or unwanted pains you are giving that. Please give the predictive data. All those rather than you testing the 1 lakh molecule on the humans or the animals, please give the short list out the 40 molecules from that. So they says that FDA is saying that, well, if I have circled it, I hope you can see it out in silico. I hope you can find it out. Right? So FDA has already, what is FDA? I hope you remember, you know that it is one of the biggest power, right? So FDA is already given the instruction, please start giving the in silico data, means use the computer power and give me the predictive data, don't spoil our resources and try to save the human lives. ठीक है जी? And no doubt software built are continuously validated and we refined these are in silico toxicology. You can look into the last corner pharmacology. In silico toxicology means you are predicting the toxicology of the given chemical by using the computers. Slowly and slowly softwares are getting refined day by day. Yes, cost and the data, robustness and the algorithms will vary from one software to another software. Let me admit that. If I am using one software free versus the paid, कहाँ 7 lakh, कहाँ free है? Don't you think the reliability of the data will vary? Yes, it will vary. But for the beginners, we always recommend to go for the free softwares. Once you get comfortable with that, shift it to the paid one. If you are really doing the research, that is going to make a big difference in the society. Okay? Certain facts and challenges while using that, अभी हमें पता ही नहीं थे, right? A lack of knowledge and awareness of the suitable AI tools of the हमें अभी नहीं पता. In fact, if I say कि even I am fully having knowledge, no, sorry, only 1% knowledge I am carrying. So whatever knowledge I am carrying, I am just trying to transfer it now. You people are at the young stage. A lack of trust, yes, a lack of trust is there. Somebody guide नहीं कहते, नहीं करना, is there, है ना? A lack of trust towards the AI or the bias towards in vitro and in vivo आता है. Overemphasis on flashy pilots at the expense of easily scalable solutions. ठीक है? Overemphasis on the flashy pilots at the expense की like I have to do this, है ना? Inadequate technical infrastructure. गवर्नमेंट फैसिलिटी है पूरी पीयू के अंदर हमारे पास 2-3 सॉफ्टवेयर्स होंगे, हमारे पास कितना होता है? प्राइवेट सेक्टर्स में जाइए, दे आर हैविंग दिस मच ऑफ स्केयर. Free online versus paid, I have already shared that. Software has its own shortcomings and pitfalls. Definition of the end user, मैं कैसे देखती हूँ, आप कैसे देखते हो, right? Depending upon the knowledge up to which एक young generation को दूंगी और एक scalable है. Then softwares in silico model might not perform well if a predicted chemical is beyond the chemical. Yes, this is one of the, like I have Dr. Rimal is sitting over here, one of my student, and I have case study from our lab also. In our case, he is working on the neurodegenerative diseases, like the target, like what is the aim of your GAN school. So neurodegenerative, he did what? He designed a library of 10,000 molecules on the Schrödinger, right? Now he did the docking, okay. He did the ADME, okay, and ultimately he filtered out somewhat 25 molecules. But we cannot ignore that what computer is giving, whether it is actually feasible in my labs also. Sometimes flashy, not flashy in this way, flashy in terms of computers also it can happen. So we need to that it is sometime beyond the chemical space. So we need to filter out, no doubt out of 10,000 he filtered out 25 molecules, and amongst 25 molecules he filtered out, we may left around 3 to 4 molecules, best molecules, because that complexity of the reaction was not feasible in our reaction. So ethical and the legal issues, whenever we people are using, आज की डेट में तो you people are using the chat GPT, you people are using the perplexity and many more softwares are there. Earlier इसको AI को क्या माना जाता था? Illegal, right? And I hope you know that now the guidelines, the journals have allowed to use this tool in case if you are quoting them as a reference. You can use them. You can use them. There is a limit. Percentage of the limit is there in the journal that you can use these data, chat GPT, perplexity or whatever the grammar what you are using. But there is a limit and the cap on the uses of these, and when you are quoting and giving the reference that I have used this from ChatGPT, you are safe. You are safe. Is that okay? So, repurposing. Shall I take 2 minutes more? Can I take 2 more? Huh? Now here is a one example in silico identification. मैंने क्या word use किया है? Identification and experimental validation, and I am giving the exercise name as repurposing. This is practically we have done in our lab. Okay? So what we did it, you people are like Parkinson? Huh? Oh, no, I am just talking about this disease, Parkinson. So, like Parkinson is there, Hana? Okay, this is the data for you people, nothing new to you. It's a neurodegenerative disease. How many percent of people are affecting? What is the reason? Pathology involved? Okay, and my target was that I got to know that me and my research partner, Dr. Sangeeta, in my department, she said ki ma'am, NF-kappa receptors are being involved in their pathology of the Parkinson, but still now in the market, though no NF-kappa is there in that. So no doubt NF-kappa inhibitors are there, but not in the term of for Parkinson, but they are present for the some another diseases. So what we did, we picked it from Medchem Express. This is again one of another database, you can write it down. This is free, Medchem Express. It is again target wise, chemical point of views wise, many point of views, it is having number of molecules in its database. So Medchem Express, we used it and we filtered out 6 molecules from that databases. NF-kappa that was showing the some good potential. So what we did, we picked the Parkinson disease target. No doubt they are the known NF-kappa inhibitors, but they have not been exploited or studied for the Parkinson. So what did we went to the PDB site and we tried to look into all these 6 molecules, and the result is in the form of the D scores, number of residues, type of interactions being involved, and jitna negative mein hota hai, the negative is the score, the better is the molecule. If you look into, you will say ma'am, Ursolinic acid at the candidate number, oh sorry, sulfasalazine. You will say what, ma'am? Sulfasalazine is showing the best results. Okay, -55 followed by -42 and so on, ardostane, and I3C is standing at the second number, fourth number. Okay, so we are interested in how many type of the residues are being involved. Mesalamine तो है ही नहीं आ रहा, normal alkyl residues आएंगे, hydrogen bonding, hydrophobic. So this was the kind of the basic information we tried to sort it out. Okay, now I told you that what are the two tools being used. Primary screening is based upon the docking and secondary screening is based upon the ADME. So we tried to study the pre-ADME. What is pre-ADME? Predictive ADME. Okay, एक होता है ADME. ADME means I am doing it in vitro or using in vivo models, but when there is a pre word, it means I have done it by using predictive data, right? So we tested all these molecules based upon their human intestinal absorption, then plasma protein, blood brain barrier, etc., etc., and we try to combine. If you look into, let me show you the one slide next. X log P. In our drug industry, log P holds very importance. Log P is what? Lipophilicity. Your drug need to have a perfect balance so that it can cross the blood brain barrier and it can reach to the target. If it be too hydrophilic, it is not will be able to cross. If it be too lipophilic, then it will cross the brain also. So it's there is an optimal value of 2 to 5 के बीच में आता है. So they said that now if you look into this score, docking score, here the first molecule was sulfasalazine. Sulfasalazine. If you look into the sulfasalazine structure, it was having carboxylic group, carboxylic group, carboxylic group. It was too polar in nature, not found to be optimal for the study. No doubt it was showing the good. Now what is the difference? How you can say, ma'am, the drug is showing the good interaction in the docking, but you are not saying it's good for the, in order to show the response at the target site, your drug should get absorbed, it should get distributed, then it should act on the target, after that it should get metabolized or get excreted. Do you think the metabolism before the drug reaches to the target is good or bad? Bad. If the drug is getting metabolized before it reaches to the target, is it good or bad? पहले हो जाएगी. So metabolism need to be studied. ठीक है. So we applied all this filter. No doubt it's showing that sulfasalazine is really 55 score, but when it comes to the X log P कितना आया उसका? 0. So we cancelled out. It's not only the one para. Docking is the static studies. Docking is static study. In case you want to support the static binding in term, एक और यूज होता है टर्म मॉलिक्यूलर सिमुलेशंस. डायनेमिक. डायनेमिक क्या होता है? स्टैटिक मीन्स ये रिसेप्टर है और पड़ा हुआ है. बिकॉज आई एम डूइंग द डॉकिंग ऑन द कंप्यूटर सिस्टम present in my body. In my body, protein is not present like that. It is continuously contracting, relaxing, contracting. By doing the supporting studies of that is called the dynamic studies, molecular simulation studies. Presently it is as us. So it's not only the docking is statics, but in order to reach from mouth to that side, it means absorption should be good. Absorption कैसे good होगी? जब HI, human intestinal absorption. So we try to, when you will come into certain market में देखोगे, have you heard about the Lipinski's rule of 5? Very good. What is that? Good. So कहाँ से आया है? साइंटिस्ट का नाम है ये क्या है? लिपिंस्टिक वाज अ साइंटिस्ट. साइंटिस्ट होने के लिए, so he was a scientist. He observed that there are market में he did research from the market that are drugs, successful drugs. He studied what is common among make them successful and make them different from the unsuccessful drugs. And he noticed that एक्सेप्टर थे, इतना लॉग भी था, इतना मॉलिकुलर वेट था. दैट्स व्हाई इट इज कॉल्ड द लिपिड drugs in detail. What were the common features that make them successful? So that if we want to become a drug, it should have these features: donor, acceptor, surface area. I hope you got it that. So that is Lipinski's rule of 5, powerful rule. उसके बाद Lipinski rule. If it is any candidate, it is having all these 5 features. That's why a computer can, do you doing one study? I will give you one assignment. You will put into the chem draw. You will try to predict the properties. A computer. But what is the logic? So out of those 240 properties, what are 5 important properties I need? Try to focus on that. At least it should have these 5 parameters, and after all these, no doubt you would falling on the 4th number according to the D score. But after Lipinski rule of 5 and the molecular dynamic studies, we reached as the 4th number candidate. Somehow, some certain time it was the reason one molecule, no doubt it was having good D. Second molecule filtered it out, it was having no doubt good score, but molecular weight was not good. Third molecule that was very costly. So I came to the 4th potential candidate, and this is how, and I validated the molecule we identified in the computer, experimental validation in our lab, and we got it published in journal. So this is how you can upgrade the level of your studies. I'm trying to slowly incorporate the acceptability rate, look into the success rate, look into the curtail of the experiment. So this is the one of the article, protective effect of indole 3 NF in silico and in vivo studies. This is our lab article. So this is I3C, and we are continuing with identified. Again I ask you, I3C. No. Did I discover this molecule? No. What I did? Identify. Now I got the skeleton from this study. Now what I am trying to do in my lab, I am trying to modify I3C in my labs. I did the modification, I got even better molecule, and we got patent on it. So this is how. So don't you think this has fastened up the reaction? Don't you think? So computers are not doing anything. Computers are helping you to fasten up the reaction. They are helping you to make better decisions. They are helping you to reduce the time, reduce the cut on the cost. Okay. I was having many more examples from our lab, but it's okay. This is a collective study with Dr. Tanzir. That this is no doubt all credit goes to Dr. Tanzir. He gave me a piece of work to do on the computers. I did the computational and synthetic part, all biological part goes to her credit, and we have many papers out of these studies. Thanks to you, ma'am.
Thank you, Dr. Nilima. We will continue